Ciao David, how are you?
All fine, I'm ready.
Okay, we have 15 people inside. I think that we can start. We are already recording.
Super.
So today we will change, and if we will speak about also some infectious disease, right? And
Yeah, but still mainly about parasites.
okay, perfect. So this is also one of your favorite topics and also my favorite topics. So thank you again for accepting and good lesson to everyone.
Thank you very much. So I am trying to share the one I hope. Yep. Okay. Just tell me if you see the slide to confirm.
Yes.
Okay, super.
Yes,
Fine.
Professor.
So, hello everybody once again. I will start with a little bit more introduction of myself because it creates a background for whole talk and I didn't do so yesterday. So I'm affiliated to two universities in the Czech Republic, Masaryk University in Brno, which is mainly research-oriented university where the parasitology is part of the department of botany and zoology. So it's really purely scientific parasitology. And then I work also for the Czech University of Life Sciences in Prague for the Department of Veterinary Science, where we do more like, I would say, veterinary parasitology and also experimental work on animal models and so on. And beside that, I was for more than 10 years a director of Veterinary Sans Frontières Czech Republic, and which gives also kind of a little bit more practical background to from the environment of Africa mainly. So this is kind of background I do. If I look what my team is doing, so we do all kind of research on epidemiology or eco-epidemiology and of wildlife parasites, domestic animal parasites and zoonoses using different models in different parts of the world. And some of these topics I will try to introduce today, but I really selected only few examples. And in the same time, I work a lot with the zoo animals, which is another topic I want to include into today's lecture, because speaking about wildlife or free roaming wildlife and speaking about captive wildlife is quite different, and both situations create really different environment for transmission of the diseases. And specifically when we speak about zoological gardens, this is very specific environment. I'll show you a few examples that really creates a challenges for veterinary parasitologists and also for, I would say, wildlife managers. And my work in the zoological garden that I started like 3 years ago, like really brought me or open myself for open questions of the proper health management of the captive exotic animals, and also kind of controversial who is responsible for that. So we see basically two ways of management of the health of these animals. There is one side that believes that basically mainly preventive measures against the parasite, or I would say control of the parasites and infectious diseases, is more part of the animal husbandry in the zoo, and the veterinarians are just coming for the more like treatment-oriented activities. Or there is another group of zoos and people that do believe that even the preventive medicine and health management is part of the veterinary care, and that the veterinarians are supposed to do so. But commonly, when I really work practically in the zoos, the result is that nobody is doing that. Because I would say the zoological staff of the zoo or the curators of different groups of animals, they do believe that the preventive medicine is a responsibility of veterinarian, And the serving veterinarian, they has opinion that the preventive medicine and all these preventive measures, it's part of the animal husbandry, and then the curators are responsible for that. And as a result, it doesn't work properly, and the animal keep dying and keep having issues because of different parasitic infections. So this is quite specific, and I'll show you some examples later. If I go very briefly into what we are doing with my team, so we do a lot of research on ecoepidemiology of diseases in large herbivores, working on helminth communities, working also on the tick-borne diseases, both in Central Europe, but now also in Eastern Africa, previously also in Levant, in Arabia, and so on, in Jordan. And not only with those, I would say, exotic livestock like camels are, but in Europe we work also with typical livestock as beef cattle, which creates another quite interesting parallel to the zoo animals and the wildlife, because more animals you keep on the pastures in the environment, closer it is to wildlife. So in my opinion, and when we speak about for example, the exotic pet, so if you look at the infectious diseases issues in really exotic privately owned pets, this is type of veterinary medicine which is very close to the medicine of dogs and cats, because these are the very well cared pet animals, usually individual animals kept somewhere by individual owners. If somebody has one iguana or one python, basically, even though this is exotic animal, the principles are close to dogs and cats. When we speak about the zoo animals or about livestock, which is kept in more or less natural environment on pastures, it's very closely related to the true wildlife, because here we usually work with the group of animals. The individual medicine is quite complicated, because even to identify the sick individuals in the big herds is complicated. So we should do definitely more the health management approaches and the herd management approaches and so on, which opens much more interesting opportunity for the infectologists, epidemiologists, and so on. Anyway, part of our research are also the tick-borne diseases, so I will give some space for vector-borne diseases in my talk, using mainly the molecular tools for description of, again, the ecoepidemiology of transmission of the tick-borne protists and bacterial infection between different species of European cervids. We work with wild carnivores. Everyone who, more or less everybody working with wildlife works also on wild carnivores, of course, and the exchange of the parasites and infections between dogs, jackals, wolves, foxes. We work with exotic birds. We did some projects on atoxoplasmosis, which is basically a coccidian infection of some starlings and other exotic birds. Relatively important part of our research is work on emerging diseases in tortoises. I'll go to that a bit later in my talk. I already presented yesterday, day before yesterday, our research on Angiostrongylus cantonensis. In Somalia, we work a bit with cheetah and parasites of these animals. And last but not least, in past 10 to 15 years, my team worked a lot with wild African great apes. We started working with chimpanzees, then we moved our work to lowland gorillas, and past 5 years, we worked almost exclusively on mountain gorillas in Rwanda and Uganda. So I have also one example from these animals. So taking all this together, these are just our field sites in Africa. So we have relatively massive experience, I would say, with different populations of great apes across the African continent. So taking all these things together, as I already mentioned yesterday, we are interested in emerging infectious diseases in wildlife. We are interested in emerging zoonoses that somehow are associated with wildlife, mainly parasitic ones, but not only. And we are interested in conservation medicine as one of the tools for the nature conservation and the health management of wildlife and so on. Fine. So speaking about wildlife and management of health and management of infections and parasites in wildlife, should definitely start, at least in my opinion, with definition what the wildlife is. And that is not really like very universal. a universal definition. But for me, the closest somehow definition of what we consider under wildlife is this one, which I found somewhere. It's maybe from Wikipedia or from somewhere, that by the term wildlife, we consider the living creatures, especially vertebrates, excluding the humans and domesticated animals, which is, in my opinion, a relatively good definition. So please, someone has to mute his microphone. We are having some voices. And when we define what wildlife is, we can jump a little bit more into what we understand under wildlife disease management. And it's, again, quite a complex, I'd say, issue or complex area that combines a range of practical obstacles logically, because whatever you want to do with wildlife is definitely more complicated than doing the same with domestic animals. But it has, and this is very interesting for me, very broad range of ethical aspects. In other words, the key question for me, and I was not interested in that much in the past, but when we started to work with gorillas in Africa, it really opened that question clearly. I'll show that in some pictures later, that how much we should interfere or how much rights we have to interfere with the true wildlife, or what do you really understand under the term wildlife? If we suppose,
Le renne devi dire maschi e femmine hanno...
Laura, please, can you switch your microphone off? Okay, let's continue. So there is quite interesting publication which is called Ethics of Wildlife Management and Conservation that I definitely suggest to read if you are interested in that. And because you study, part of you studied wildlife management, so then it's should be something, in my opinion, you should be interested in. It was published in 2012, and it, of course, defines that when we speak about the wildlife, is for most of us as for humans, it has relatively huge instrumental value, which basically means, if we simplify that a little bit, that we can consider wildlife as a resource, but we can also consider wildlife as a threat. And the threat can be direct, which means the brown bear is coming eating you. The threat can be from wildlife towards, for example, the domestic or to domestic animals. But the threat also is having wildlife as a source of zoonotic infections. So this is very instrumental way how to classify that. It's quite a rational way. But there are also many other aspects that are more ethical and more difficult kind of to discuss, or more complex, including the well-being of the individuals, because we should consider that wildlife, it's a population, or animals living in some wild population, but the population also consists from individuals. And we should also consider that many of these species are protected species. And we also, so this is the level of the species, going from individual to species or population. But we should also consider that all these animals are also part of some ecosystems, and we also tend or try to protect the ecosystem as such. And protect the wilderness as an asset. And if we jump into this, and if you really think about that, it looks, at least to me, that we are a little bit losing the rights to interfere and to do the management. Because if we want to protect the wilderness, it means we do not interfere. And that is kind of huge dilemma that we have, and it's mainly dilemma for future wildlife managers or people involved in the management, which are exactly you who are listening. And the question is, how much shall we interact? How much rights we have to interact, to manage? Because more we are interacting, more we are managing the wildlife, more dependent the wildlife is on us, and less wilderness we have around us. Yeah. So it's, as I said, it's not truly a question for a parasitologist or infectologist, but it's a background on which we should consider all these aspects. And it's, in my opinion, good really to think about it, and sometimes maybe even rethink our approaches, because there are many examples that more intensively we try to manage the wildlife, worse the situation is. So if we go back to that study I mentioned that I truly believe that it's interesting for you, there was kind of conclusion of so-called balancing concerns, saying, which is logical, that first of all, the management and usage of this wilderness creates many ethical disagreements and dilemmas, and logically different groups, different stakeholders, different opinion, whatever makers can show different preferences, different interests, different concerns about individuals, populations, and the value of this ecosystem. And depending on our way, how we prioritize these values, it determines the policy, how we want to manage the wildlife, including the diseases. Yeah, so if you prioritize the human health, then technically speaking, it gives us some rights to manage the populations of wildlife the way we need to protect our health. But taking from the opposite side, if a perception of other people is that we should prioritize the wilderness as such and the well-being of wild animals, then of course we are losing the rights just because of our health to start interfering with them. Yeah, and this is the debate which is endless, and this is the debate which is good to keep in mind, and at least to, as I said before, at least start thinking about it. Because the fact that we are veterinarians, the fact that we can diagnose the disease, doesn't implicitly mean that we have rights to interfere with wildlife and to start organizing the nature the way that we want. No. So, protection of wildlife, it's obvious. And at least in the Western society, people generally tend to believe that this is something that should be involved in or implemented into our culture, and basically is implemented our culture. Also with many consequences that I will speak about later. But in whole Europe, or in most of the Europe, we are witnessing so-called rewilding processes, which are sometimes spontaneous, sometimes they are a result of our positive attitude towards the nature. And as a result, it brings this process of rewilding, brings the animals closer and closer to humans, in many situations in higher and higher quantities, which creates quite interesting interface for the disease transmission and parasite ecology and so on. And it's, in my opinion, one of the challenges for the future wildlife management, or even for current wildlife managements, to balance the positive aspects of having wildlife close to us with the negative aspects that this brings. And it's not only about the carnivores, it's also about the herbivores. Here in Central Europe, we are witnessing like overpopulation of the landscape with wild herbivores. The population of the red deer was never in the history as high as nowadays. And it creates significant source of some pathogens. It has impact on the dynamics of the tick populations, tick-borne diseases, some other parasitic things, and so on. Yeah, so this is about the animals, but we should also consider that the parasites themselves are part of the biodiversity, and if we do agree that we protect the wildlife as a part of the biodiversity, basically it mandates us to protect also the pathogens from the extinction and to protect the pathogens as part of this biodiversity, which has again many theoretical, it's like playing with the words a little bit, but it has also many practical implications. So this is one reason why we should care about the parasites, like about, I don't say really protecting them, but caring about parasites as part of the ecosystems. But we should also keep in mind that if you look at that as evolutionary biologists, the parasites, they really have true importance, and we can say positive role in the evolution of hosts. Because of the arms race, that is well-known mechanism and so on, like basically our parasites make us stronger and better in next generation because of selection procedures and so on and so on. That's clear. So we cannot evolve without being confronted on the almost daily basis with the pathogens. But in the same time, and it's more and more clean, the parasites themselves, and it has implications for the zoological gardens. The parasites themselves, they have real importance in the ontogenesis of the immunity. In other words, and this is the big dilemma we have in the zoos, if we clean the parasites from the animal population, which we are trying to do in the zoos, and we have good reasons in many situations for this, or many instances. But if we clear these animals from their parasites, we cannot be surprised that these animals are not really having functioning immunity, which again creates a lot of dilemmas, a lot of, I would say, internal conflicts and tensions, specifically in situation, I will not speak about it that much later, so I'll say that now, specifically in situations when we keep the animals in captivity as a possible genetic resources for reintroduction these animals to the wild. Because there are many practical examples that this doesn't work. Because if you keep the animal in a zoo, the nice example are wild Przewalski horses. Czech Republic is really famous for having huge captive stock of these wild horses, and they are gradually being exported to Central Asia, released into the reserves in Mongolia and so on. But these animals keep dying in the natural ecosystem after being released because they do not have immunity. During the way we keep them in captivity doesn't give them opportunity to mount, for example, immunity against tick-borne diseases. And then you release these horses to the wild, and within half a year, they die because of equine pyroplasmids. And I'm not saying that we know how to manage these, but we should keep this in mind, that having the animals without parasites in captivity creates quite an artificial situation for the hosts themselves. And I'll show you some examples later, maybe. So there are another studies that deal with this beside the one I mentioned. So if you are really interested in that, go for it, read it, because at least for me, it was kind of really interesting reading. Because I'm parasitologist, I'm partly veterinarian, but mainly parasitologist. So I'm not really the philosopher thinking about the ethical dimensions. But that's bad, because if we really do something with the animals, we should always think about the ethical dimensions and about the management and the consequences of this and so on. So go for that, read it. I truly recommend. So how is it in the zoo? So far I spoke mostly about the wild animals, but the zoo itself, and I'm supposed to speak about both in my talk, which it doesn't go really well together. The zoo itself, it's a really specific environment. It's one of the most complicated environments for the epidemiologists or the animal health managers. Why? Because the zoo brings together, this is a map of one of our zoos where we do the diagnostics. That doesn't matter. It doesn't matter that it's in Czech. But all things that I want to discuss are clearly seen here. The zoo is semi-natural environment full of animals of absolutely different origin in extremely high population densities, mixing all types of animals you can imagine, which creates truly a kind of whirlpool of pathogens that are being transmitted, parasites that are being transmitted, ideal environment for many aberrant infections. So whatever is working normally in the wild populations works really differently in the zoo. I'll show you again some examples later. Other real aspects of the zoos, this map of another zoo, is that beside the animals that we keep, there is a lot of other organisms that are interfering with the dynamics of the infectious diseases there, including parasites, doesn't matter, it's the same. We have the feral, wild, semi-wild, and domestic cats in almost every zoo, including the zoos in Italy, where I have a chance to work. But we have also wild carnivores coming to the zoos. In basically every zoo in the Czech Republic, there is 1, 2, 3 more foxes living in or around, including the badgers. And martens and other wild carnivores, and they are again interfering with the cycles of the parasites. Of course, we have population of birds. I have here symbolized that only by a blackbird, but every zoo has population of tens of species of the birds. Some of them are stationary, some of them are semi-migrant, some of them are migrant, bringing loads of pathogens, but also able to take loads of pathogens out of the zoo, which is another risk. Then we have synanthropic rodents, which are in many zoos creating the real issue as a source of infectious diseases, including, again, some interesting cases from Italian zoos that I have no time to mention, but they are there. And then in most of the European zoos, at least in the Czech Republic, the dogs are allowed to come together with the visitors, which creates another quite specific aspect of the. I'm definitely against that, but unfortunately, this is the culture of living with the dogs in Western Europe or Central Europe or wherever. Yeah, so we are allowing the dogs coming to the zoo. Of course, they are not in direct contact with the animals, but they are bringing some of the pathogens, and they can also take, to be very honest, some pathogens again out of the zoo to the domestic dog population. So that's quite complicated. And to make it even more complicated, of course, every zoo is a habitat for disease vectors, mosquito, for disease intermediate hosts, and so on. So this is quite complex environments. It's not wild nature. It's more complex than the wild nature. If you feel that it's simplified, no, it's not. Because the normal nature next to the zoo evolved through some processes of coexistence. And basically the European ecosystems, they have a history of, at least from the glacial period, so I would say tens of thousands of years history of coexistence and creating the rules, creating the relationships and so on. But in the zoo, everything is new. And we are changing that almost every second week. We are bringing some new animals and we are sending the animals outside and so on and so on. So this is really complex and not that much related to issues we see in wildlife, but it's overlapping with the issues of the wildlife. So, and many times quite frustrating environment to work with the diseases, to be very honest. Yeah, we have also synanthropic insects, of course, in the zoos, as I show you. So that was part of the background in which we discussed the parasites and infectious diseases. So in most of my talks, I start with a saying, which I still feel it's important baseline for our discussions, that having parasites, it's a normal situation. Having parasites themselves, it's not a disease. It's not something abnormal. It's not something alarming. And so it's normal. Because the word normal is an attribute of majority. And if you look to the animals, any wild animals around us, absolute majority, if not any, if not all, sorry, have parasites, which means having parasites, it's normal situation. And having parasites as such definitely doesn't create an imperative for management of these parasites. That's not the reason. And also, presence of the parasites is not a disease. We should really distinguish when we speak about the management of this, what is presence of the parasites and what's parasitosis, because it's not the same. Yeah. Parasitosis is a clinical perception of us as veterinarians or animal owners, but having parasites, it's a natural process. And sometimes having parasites can result into a disease that we call parasitosis. But in many other situations, having parasites is not causing a disease, and then it's not a target, when it is not zoonosis. It's not a target or aim for the management. But why to care? So why we should care if it is normal in wildlife to have parasites? There are several reasons why we have to care. And there are several, we say, exciting even opportunities and in the same time challenges for you as future generation to care. First of all, because of zoonosis. Part of my talk will be about zoonosis. And when we speak about zoonosis, it's changing the story, of course, changing the game. Because we don't want to see us dying or being seriously sick because of any zoonosis. We don't want to see our children being sick. We don't want to see our relatives dying because of the zoonosis from wildlife. So that's a serious reason to care about zoonosis, care about parasites and pathogens in wildlife. But then we also should consider that a lot of wild animals nowadays around the world, including Europe, are endangered animals which are anyway threatened by other factors, by other stressors, by changes in the environment, deforestation, hunting, poaching, and so on. and their populations are fragmented. Their populations are very fragile and not stable. And then logically, pathogens that are coming into these populations represent another threat, specifically when these are non-native pathogens, and can really drive the species to the extinction, even in well-protected areas. This is quite specific aspect of emerging diseases in wildlife, that even though you protect, you keep the endangered animals in highly protected reserve, there is a pathogen coming and all the animals can die, and 50 years of your attempts to conserve the animals, to protect the animals, all this is lost within one epidemic of some viral disease. And of course, this is also my perspective, the parasites or infectious disease in wildlife is a very interesting research target because of all these reasons we mentioned before. And more complex we try thinking about it, more interesting it's becoming, but also more difficult it is. So studying a single parasite in a single animal is easy. This is what we do for more than 100 years. Studying communities of the parasites in communities of the animals, it's definitely more interesting, but definitely it's more challenging. And this is somehow the level that we now reached. We try to understand, but what we still are not reaching, understanding the whole picture. The understanding the ecology of the ecosystems, the hosts and the parasites in the dynamics of time and so on. And once we include also the human society, which is more or less the anthropology, then we really are getting the full frame of the management of the wildlife diseases. And then it's becoming almost holistic and very difficult level of even thinking, and then practically very difficult level to reach. So, as I said, having parasites is a normal situation in all kind of, basically, I would say all kind of vertebrates. Every single species, and probably in the wild, every single individual, maybe even in captivity, should have some parasites and has some parasites. Regardless of these are the exotic animals, regardless, this is the wildlife living either common or rare around us. All of them, they have parasites. So we can really consider having parasites is a normal. But there are many buts behind that, specifically when we speak, as I said before, about fragmented populations, captive populations, artificially managed populations. And this is what my talk is about a bit. And because we don't want to see this. Is this normal? Yeah, we already agreed that for a carnivore, having roundworms like Toxocara is normal. And I definitely say, yes, it's normal. But is it normal that young fox, like the one on the picture, dies for such an infection? We can say it's common. It's probably rare in the wild, but it's common in captivity. So this is not normal anymore. And we can ask, okay, so why this is happening? What's not normal? Not normal in this case that otherwise healthy animal is dying because of abnormally strong infection intensity, but by parasite, which is considered common and normal. And the answers are a little bit in the area of transmission ecology and the principles of the infection by different groups of parasites. It's really simplification, but for me, I see basically two major, when we speak about parasites, or even pathogens as such, but let's focus more on parasites, we can see basically two lines of how the parasite quantity and the impact of parasite on animal health depends on the overall situation. In most of the situations when we speak about the helminths, the infection intensity of helminthosis, like we see on the picture, truly depends on the environment and the lifestyle and of the given individual. First of all, because of some encounter filter, ecologists, they call it encounter filter, that first of all, the animal should be in contact with the given pathogen. And this depends with the stages. And it, of course, depends on the lifestyle, depends on the ecology, depends on the presence of the vectors, depends on the presence of the intermediate hosts and so on. And all this creates the quantitative aspect of the infections. And what's important is that specifically in parasites with direct transmission, which is at least in part also our Toxocara, which is on the picture, this depends on the population densities, because the transmission of these parasites and contamination of environment by these parasites, it's density dependent. And this is one of the major things that answer the questions, what's wrong in the given situation that this particle of baby fox died because of Toxocara. So even though having the parasite is normal, being enclosed in captivity in a place which is just basically few tens of square meters is not normal for a fox. And this is exactly what's happening for most of the animals in captivity. And this is happening, of course, to the livestock as well, that we keep these animals in too small space that is then becoming, of course, over-contaminated by the infective stages. And each infective stage, each egg of Toxocara can theoretically, or larva, can theoretically be responsible for an adult worm in the intestine. More eggs the fox is eating, more worms the fox has in the intestine. So it's a relatively straightforward mechanism, easy to describe, but very difficult to manage in the environment. And we can see these also not only in captive animals, but even in the wild animals, because logically, with increasing population, and it can be protected species, or maybe not even protected, but with increasing population of given host, the landscape for the parasite transmission is changing, the contamination of the environment is changing, and the infection intensity that were normal some years ago can really go high, starting to create an issue for the population as such, which is important also for the endangered animals. I will show you later. So this is the one line, which basically depends on the densities, on the vectors, on the ability of the host, or the probability of being confronted with the given pathogen. And then we have the second line, which is true mainly for the protists, but many viruses as well, and bacteria and so on, but also for some helminths, for example, strongyloides. And these infections are immunity dependent. So there is a huge difference, because in case of Toxocara, if you are infected with one egg, one worm develops. If you are infected by Giardia, which is an example on the picture on that I showed now, if you are infected with a single cyst and your immunity is not working due to endogenous autoreplication or replication of the parasite, the infection gets very fast. The high intensity can having the impact on the environment. Can have impact on the host health, but it's not dependent on the environment. It's not dependent how much the environment is contaminated by cysts of Giardia, because they can reproduce very easily in the host if the animal is not okay, not having the properly working immunity mechanisms. And for me, this creates really huge changes in the management. Yeah, because if I see the fox having a lot of Toxocara, the first question should be, okay, what's wrong with the environment? It's a disease of the environment. It's not a matter of the fox. Something is wrong. Something is wrong with the population density. Something is wrong with the parathenic hosts, or highly infected parathenic host. Something is wrong with the contamination if it is captive one. But if I see the fox having the strong giardiosis, then I should ask, okay, what's wrong with this fox? Why this particular animal is having a severe diarrhea caused by otherwise normal parasite, which is present everywhere? And at least for me, this is huge. Yeah? This is a really huge difference, and that should be considered. So we are back to our wild animals. And one thing that we should consider, that what is normal, I hope it's obvious already, but I'll repeat that with some examples. What's obviously normal for a wild lynx somewhere in Italian or Carpathian mountains is not normal in a zoo, because everything is different. And what's mainly different is the space. And logically, while the lynx in the wild is having mainly heteroxenous parasites, which are transmitted by its prey, the lynx in the zoo is affected mainly by the parasites, this direct transmission, which are transmitted through the environment and can very fast reach the level of contamination of the environment, which is basically a killer for the captive animal. Yeah, so this is quite a huge difference. The same I show you right now, the practical example. The same is, for example, true for a wolverine. The large, I took it just an example from our practice in the past months. Large carnivores living in North Europe, north of the United States, and so on, subarctic areas. Huge animal resembling the huge marten, you probably know it. Living in the areas of tens of square kilometers. Extremely huge home ranges. So this is the wild. This is the captivity. Even though our zoo is perfect, it's not our zoo, it's an example. I mean, the zoo. Even though the zoo is very good, even though the exhibition is wonderful, I would say this one is really not bad. The space the animal has is nothing. Yeah, compared to the home range in the wild, it's absolutely nothing. Yeah. And this is it. So the very basic or common parasites in the wild are suddenly becoming an issue in the captivity. And we should also consider in the management of captive animals that this is not only the wolverine having, for example, uncinaria, which is transmitted through the environment, like, you know, the life cycle of the strongylids. Yeah. And we also know that some of them, they penetrate the skin. And it's really different if you live somewhere in Alaska, walking tens of kilometers in more or less sterile environment where the parasite is not present. Or if you live in a cage, which is otherwise nice, but contaminated by the feces from which the larvae are emerging on the daily basis. And you are stepping every day, basically 24 hours a day into the soil, which is contaminated by larvae. Logically, the level of the infection is enormously different. But we also have the vectors, of course, in such an environment, as we mentioned, and we also have the intermediate hosts. And all these are the factors that we should consider if we truly want to manage the parasitic diseases in captive wildlife. Yeah. So consider making kind of conclusion of these things about the different types of parasites with different types of life cycles and so on. We can conclude that, and different pathogenicity, that not everything what we see or detect during the parasitological diagnostics is having the same importance, considering the condition in which in the given time the given individual is living. Yeah. Because in practice, usually, when we really ask for the results of the parasitological examination, if we really do the methods, which are basically simple, if we really do the parasitology, all is simple. Either we look into the shit, we look into the blood, we use the microscope, we use few methods, some PCR, some serologies. It's simple. It can be done by machines almost. But what to do with the result? This is the question for a veterinarian or the wild manager. And that's already complicated. This is just an example of normal parasitological result of a monkey from one of our research. It was Agile Mengabi in Central African Republic. But looking at that, it's already showing really huge complexity. If we start from the top, going through these pictures, so the first thing under number one is apparently an amoeba. If you are more into that, we can say, even though we did parasitological examination, this is Entamoeba coli, which is not a parasite. It's a commensal, so we can cross it. Yeah, and we can say, if we have knowledge big enough, we can say, yes, this we don't consider. If you look to the number two, Iodamoeba buchli. Again, if we are aware of all things behind and around, we can say, yes, this is also not a pathogen. Let's cross it. Yeah, it's a commensal. Then the number three is interesting, because if you look at that and if you are a little bit into protozoology of animals, you say, oh, that's Balantidium. We have Balantidiosis. And that was what was believed until relatively recently. But thanks to the intense research, thanks to the molecular tools, we know that this is not Balantidium in case of this Mangabe. It's a Buxtonella ciliate, which is having the same cysts as Balantidium. But again, it's considered not pathogenic. So this is what we see here in this animal. It's not Balantidiosis. No, it's presence of Buxtonella, undescribed species of Buxtonella, which is not a pathogen. So let's cross it. And this way we can basically dissect the results of the parasitological examination. But doing that, we need enormous knowledge about the given species and about the context in which we are examining the animal, which is challenge. That's definitely not easy. Yeah, we are back to our Wolverine, the case of single individual that was recently imported to our zoo from UK. First thing that was interesting is that the animal came with negative results. So you would say, perfect. Colleagues in UK, they did great job sending us the animal, which is perfectly okay. But as a normal daily routine, we performed the parasitological examination and the Wolverine was not really negative. It was having quite massive infection with 3 or even 4, one of them is not shown here, 4 different types of the parasites. Starting from the right, Giardia. You probably recognize the cysts if you are a bit trained. Yeah. Massive giardiosis. Again, opening the question, okay, well, what's wrong with this Wolverine? Maybe it's stress, maybe it has some chronic immunity dependent processes, but something is wrong. The picture in the middle, Uncinaria stenocephala, the one we already discussed. The picture on the left, you easily say, oh, these are larvae of some lungworm. Massive infection, also not good. And that was true. And this is the result of the examination, but it doesn't give us yet the hint how to manage the situation. Because considering the Giardia, we should probably use totally different way of thinking and approaches than having Uncinaria in the middle or having the worms. And this is the lungworm, sorry. So this is the first level of the diagnostics. Of course, sorry for check here, but to diagnose the lungworms, first of all, we should know that we should apply the larvoscopic method. If the laboratory is doing only the flotation technique, then it would detect easily the Giardia. It would detect easily Uncinaria stenocephala in the middle, but with high probability it will overlook the infection by the lungworm. So first of all, we need to use the proper methods for the diagnostics. But then having the larvae, sorry, again, it's in Czech, but doesn't matter. Having the larvae, I just wanted to say that then we can look as parasitologists, we can look morphologically and try to identify that, which is already quite high level, to be honest. This is not a routine. Yeah, routinely, in the very routine laboratory, this is not done. But this is what we can do. We can try to identify what the larvae are. And if we perform this, we use different, basically taking this example of particle situation and the management of particle situation, we have at least 3 layers of the diagnostics. From the top, the first one is very superficial. Okay, we see nematode larvae, maybe the lungworm larvae. What it means for the management? It means we should think about the treatment, of course, but for the rest, we know nothing. It's not enough. We need to go more deeply. So we do morphology. Yes, perfect. We identify this is the middle level. Yes, this is nematode larva, probably Angiostrongylus. Wow, okay, it shows us more. But still, this is not enough to really set proper management. We need to understand more. So then we have the molecular work. We do PCR, we do sequencing, we have the final sequence, and we have confirmation. Yes, this is Angiostrongylus vasorum. Perfect. So at the level of diagnostics, satisfaction. We have massive infection of Angiostrongylus vasorum, and we should start thinking about the treatment. But this is not what I want to discuss. The treatment was finally done, and finally it was managed. But I want to ask how this exercise helped me regarding the management of the animal. We should consider that the animal is living in some cage. And we already know that the cage is contaminated by eggs of directly transmitted Uncinaria stenocephala. But we know that also the cage is contaminated by snail-borne Angiostrongylus vasorum. And we know that we really cannot exclude the presence of the snails in the cage. So we know that probably we are already having the infection at the level of intermediate hold, which means even though we treated the animal, we should expect that the animal sooner or later will get infected again, which means we should start monitoring it. So it means that so the first, after the treatment, after we are sure that this poor wolverine is negative, finally, having no worms, no larvae in the feces, this is not the end, it's the beginning. We must be sure by repeated examinations of the same individual, I would say in 3 months interval, we must be sure that the infection is not coming back. And this is still not the end of the story, because we know that mollusks, specifically these slugs, they are quite mobile animals. So they can easily go from one cage to the another. They can go in a night, they can go 50 meters. There are other animals. Or they can go other way, where are, again, other animals, and so on. They can infect even our wild foxes, which we have in the zoo. So suddenly we have a situation that because of importation of one individual from UK, we theoretically created a hotspot of Angiostrongylus vazorum in the captive environment of the zoo, which can be a headache for next decade. Maybe it will not happen. We can hope this is not happening. This is not ongoing, this process. But in the same time, we should be aware of the risk that this can happen. And we should implement that into the management, which also means we should start probably more frequently in a coming year, examining the susceptible host for Angiostrongylus vazorum, sorry, vazorum is vazorum, for Angiostrongylus vazorum, by larvoscopic method, not only by the flotation, as was done more or less until now, but since now we have to start also the larvoscopies to be able to detect this one. And this is already something that we can consider management. But it took us 3 months. We examined something between 50 and 100 samples from the same individual. We implemented molecular work and the sequencing. And as a result, we can say, yes, we know what's going on in this particular animal and in this particular cage. Problem is that it's a lot of effort, that it's in many other situations people are not ready to do or undertake, or they are not willing even to invest that effort and that money into solving the situation in one individual. But then we cannot be surprised that the zoos are endlessly a source of the problematic situations with parasites. Okay, so that was just an example. We'll go to similar examples a little bit more. We still have plenty of time. I promised to speak about emerging infectious diseases. To say what the emerging infectious diseases, specifically with zoonoses, is after the COVID-19 pandemics, it's relatively easy, and I will not speak about COVID that much because it's already over, we believe. But I want to remind you about the definition of emerging infectious diseases. By American CDC, the emerging diseases are defined as the diseases that suddenly appear in a population, in our case of wildlife, but the same can be in livestock or humans or whatever, or the diseases with steeply growing importance or occurrence, which means the disease that is really showing changes in the dynamics of its incidence and so on and so on. So either it's absolutely new, or it's really, I would say, spreading fast and having huge impact. We can ask, okay, why this is happening now? I will go very, very fast through that because it's well known. We can conclude first, it's not happening only now. It was here every time. But nowadays, the emerging infectious diseases are an important part of the global change. And it's one of the maybe, and COVID-19 showed us, we got a lesson that it's an important part of the global change. And that the global change is ongoing or is here, there is no doubt, of course. And we will, I can almost promise that the new epidemics are coming. But it's not only now. Like if we go to veterinary medicine and to wildlife and these aspects, probably the first really well-documented epidemics of disease that was truly emerging was introduction of rinderpest into African continent in 1889. So it's a long time ago. And what's important here that it really happened, other important thing is the speed, the disease, even in that time, which was definitely not the time of globalization, but maybe it was already a root of globalization because the virus became global. And it took only 10 years for the virus to pass 5,000 kilometers. But we are speaking about 19th century. Nowadays, to pass 5,000 kilometers is a matter of days, and it was showed. We are shown nicely by COVID-19 and its spread. Yeah, so the global change is not, or it was like the epidemics or the emerging diseases were happening before, but now we have the environment in which they are happening much faster. If you look to the list of emerging infectious diseases, this is a slide that I keep for, I would say, maybe 15 years in my talk about emerging infectious diseases. And if you look at that, and if you look to the red numbers, in years ago, it's more than, it's 20 years. No, it was not, but doesn't matter. It's a long time ago, I put SARS there. You know that it's a coronavirus infection originating from carnivores transmissible to humans. I recommend well the pandemics of SARS, mainly in Southeast Asia. So it was 2002. Then, when I was making the later versions of the lecture, I put also MERS coronavirus, which was 2012. And I already proposed, as you see, and many others. And in 2019, I had to add another line. I almost do not have space on the slide now, putting SARS-CoV-2, or we call it COVID-19, in 2019. And sooner or later, probably, I will anyway put another line, putting there another coronavirus that's considered emerging in human population. So it's a historical slide, but it shows some principles. In animals, it's more complex. In animals, we can say that nowadays we have hundreds of emerging diseases that are really considered emerging and that are really having impact on the populations of wild animals. I selected only few just to show the diversity of the hosts and the We have chytridiomycosis, for example, amphibians. We have a range of diseases in avian hosts. Now, avian flu, or influenza is absolutely emerging issue, alarming emerging issue in wild birds across Europe. You are probably aware about that. Yeah, there are tens of thousands of cranes dying, seagulls dying. And so I see it in the Czech Republic, population of peregrine falcons destroyed by avian influenza and so on. So the birds are seriously hit. But as another example, I have there mycoplasma in the sea turtles. We have also some viral infections of sea turtles. We have a huge group of mammalian morbilliviruses, but we also have diseases of corals, infectious diseases of the coral reefs. We have henipaviruses as a group of viruses associated with fruit bats, and so on and so on. So basically, any group of wild animals we find a disease that clearly shows the patterns of, or fits the definition of emerging disease. And in absolute majority of the situation, this is part of the global change. So not only the livestock diseases, not only the human diseases, but also the wildlife diseases are part of that. And unfortunately, it can have detrimental impact on the biodiversity, and it also showed such an impact in many circumstances. Yeah. So if we go a little bit deeper into reasons for the emergence of these infectious diseases in wildlife as part of the global change, we can say it's a complex issue, there is no doubt. But this is quite artificial grouping, but I keep it in this speech for ages, and I still feel that it works, like for understanding these reasons, even though they are overlapping. But one of the bunch of the reasons are associated with ecosystem changes. We can say alteration, but maybe better to say ecosystem changes. This can be at the global level, this can be at the local level, but the ecosystems are changing, there is no doubt. But as a second reason that fits, or it's true for other pathogens, is that the global change facilitates the speed of movement of the pathogens, either alone or together with their hosts. So organisms are simply moving. They were moving always, like the migratory birds and so on, but nowadays they are chaotically moving all around the planet. And then we also see the changes in the evolution of pathogens, and we see it repeatedly. It's not only COVID-19 and related viruses, but For example, if you go to parvoviruses in carnivores, we see the evolution. There were the new variants of the viruses appearing every few decades, being more pathogenic or showing different pathogenicity for different group of carnivores. That's the evolution. And then we should be also aware of the fact that these methods, new methods of the pathogen diagnostics, we of course are detecting more pathogens, which is not truly the emergence. It's more or less emergence of the interest. And specifically after COVID, but even before, connected to Ebola and all these things, we are really searching for the pathogens. Even me in my research team, we are more or less hunting the pathogen down, like to find the new ones and to prove these are pathogens and so on. So that's a little bit artificial, but still it's part of the story. And if what I said before, it's overlapping. So in case of COVID-19, and this is the last time I speak about it, we see all together. There were at the beginning ecosystem alteration, direct contact with humans, with some wildlife, even though we don't know exactly how this happened, it happened because of the ecosystem changes. It's not natural that the humans are so much in contact with wildlife. So that was artificial, and suddenly, accidentally, that happened. And then what's the next? Of course, the movement of the pathogen. Because of the globalization of the world, the pathogen, it took 3 months, or if I remember well, the virus to fly around the globe. Changes and evolution, sure, because during the pandemics, we saw different variants of the virus, and their emergence was of course driven by the global distribution and fast movement. But we were also facing, logically, the novel methods of the diagnostics. It was so fast, it never happened before. Such a fast development of new diagnostic approaches never happened before. Yeah, and so one virus, one epidemics, all factors of this emergence being seen together. But let's look for some more examples from the wildlife. The ecosystem changes. They can be, of course, at the global level. Typical example is the global warming. Yes, it's a change, it's environmental change, and it has impact on spreading of the diseases, spreading of the vector, distribution of the pathogens, and so on. I don't like this aspect that much because it's very difficult to discuss. Basically, we can connect anything to global change, to global warming, because simply global warming or climate changes are happening. But they are anyway often combined or mostly combined with the other factors. And sometimes it's not easy to distinguish what's the primary or what's the secondary factor for the emergence of given disease. So let's look a little bit more to the local environmental changes in the ecosystem, because this we understand more and they are easier to describe. So basically, for anyone who is, so basically for all of us, even the very basic knowledge on the ecology logically shows or lead us to the statement that any changes in the ecosystem can, of course, have impact on the changes in the pathogen circulation. That's no doubt, because the pathogens, as we said at the very beginning, pathogens are part of that. So if you change the system, logically, a lot of change also for the pathogen transmission. So in this context, we should consider that not only the habitat degradation, which means like the negative changes in the environment, impact on the parasites and pathogens, but also the positive ones, considering also these rewilding processes that I already mentioned. So if we are trying to protect the nature, if we are bringing nature closer to us, no surprise that new opportunities and new conditions for the disease transmission appear. There are quite a lot of examples in this, like Lyme disease in USA, mycoplasma in birds, trichomonas in birds. I'll give you examples from mountain gorillas, but really the true textbook example is, for example, the infection in the sea otters and so on. As a documentation of how much the rewilding processes and changes in the population of wild carnivores can impact on human health and domestic carnivores, we can take a case of red fox. This is the carnivore which is now, at least in Central Europe, really widely discussed. Because after eradication of the rabies by oral vaccination, its populations are growing, and because the societal changes the foxes are becoming urbanized, living closer and closer to the humans in higher and higher population densities. And we should consider that the fox is an important host of the parasites. Here it's not complete, but here is a list of parasites that foxes share with the dogs. So it makes a real difference if the dog is living in the environment together with foxes, or whether the dog is living in the environment without the foxes. And the red ones are zoonotic. Again, it's make a huge difference for the human population if we are living together with the abundant fox population, even in the cities. And this is the ecology, because the population densities of given host in given ecosystem, it's a part of ecology in this ecosystem. And the fact that the population densities are growing is a reflect of some ecological changes. And logically, I'll speak later if we have time about Echinococcus. The ongoing epidemics of Echinococcus multilocularis is nothing more, nothing less than an impact of ecological changes in the populations of the wild carnivores. But the case of the textbook, or from the textbooks, is infection in sea otters in California. Even though it's in all the textbook and possibly many of you already heard about this topic, I still keep it in the lectures because I like it, because it's quite, or documenting quite a global issue. For those that you don't know, the sea otter, I mean the southern subspecies of the sea otter, Enidralutris nereis, it's a large marine carnivore living along the eastern coast of the United States. It was really driven almost to the extinction. But in the second half of 20th century, thanks to the strong collaborative conservation effort, the population of sea otters got kind of stabilized and started to grow until the 90s, where there was again significant drop of the population caused by the direct mortalities observed. And what was interesting that we speak about the 90s of previous century, it was the era of very well-developed wildlife medicine in the United States. So it was not that difficult to discover what are the causes of this population decline and why the sea otters are found dead on the beaches. And it was recognized that there are two protozoan pathogens involved, Toxoplasma gondii and Sarcocystis neurona. Both are the infections of the terrestrial animals, of course. So how it comes that suddenly the sea otter, the marine mammal, is dying because of these terrestrial protozoans? The answer is somehow in the life cycle of Toxoplasma gondii that you definitely know. I don't need to go to that. But if you look at the whole life cycle, it's apparent that if the sea otter is somehow infected, this should be by the oocysts. Because sea otter is not eating neither the pigs, nor porks, no mice, no humans. Yeah, so no, the oocyst is the stage which is responsible for that, and it was relatively fast by series of experimental and all kind of, I would say, all approaches combined together revealed that yes, the reason is that the seawater, at the local scale along the American coast, but also at the global scale, is contaminated by Toxoplasma gondii. Question is why this is, and we can more or less only hypothesize that, even though there are some nice models for that. And we can say it's mainly because of, first of all, the ecology at the continents of the cats. Populations of the cats, in some reasons, the domestic cats are, and feral cats, not the wild carnivores, because they are basically disappearing very fast. But the domestic cats are replacing the wild carnivores in many parts of the world. And in the same time, what we see are enormous erosions. So enormous amount of the soil and the organic material is being brought to the global sea by the water, connected also to the weather changes, connected to floods, connected to absence of the mangroves, absence of the swamps on the rivers, and so on. So simply the ocean is becoming contaminated by Toxoplasma. And it has impact not only on the sea otters, but it has impact also on the dolphins, on the whale, on the beluga whales, and so on. Yeah, so it's clear. So this is environmental change that is causing, not Toxoplasma, that is causing the mortality in the sea otters. If you look to the other parasite, it's even more interesting because it's Sarcocystis neurona. Sarcocystis neurona is, I'm sure you are aware about that, but it's associated with possums. It's a marsupial living on the North American continent. Again, nothing aquatic. And in this case, there are two aspects combined. First of all, the possums are, again, contaminating the environment, which is more or less natural. But the fact that the sporocytes of Sarcocystis neurona are going with in high numbers to the water is already kind of artificial. But there is one more interesting thing is, can be seen in the map of distribution of possum. So you see it on the left side. So the distribution, the current distribution is green on that. And you see that there are basically two areas, the eastern and the western. The eastern, the main one, going along the east and south of the United States to Central America, this is the native distribution range. So this is where the possum was distributed always. If you look to the western part of the United States along the coast, it's a population that was introduced in the 18th century by humans there during the 1819, during the colonization of so-called Wild West. So basically those were the farmers and other people coming to the west of the American North American continent, bringing the possums with them. The possum established the population alongside the whole eastern coast of the western coast of the United States. And this is exactly where the sea otters are living. It's exactly matching the distribution, even though the possum is on the land side of the coast and sea otter is on the ocean side of the coast. Still, this absolutely new interface. So it's ecological change combined with the movement of the host of the new pathogen. So it's an introduction of a new pathogen into a new ecosystem together with the host. Speaking more about the sea otters, the other reason that significantly contributes to mortality of the sea otters is presence of acanthocephalans. And what we know nowadays is that these are the parasites, the helminths, which are primarily associated with aquatic birds. So the normal definitive hosts of these acanthocephalans are the aquatic birds. We can again say, okay, but the aquatic birds were living always close to the sea otters. Yes, they were. But we know that only some types of crustaceans are important intermediate hosts of acanthocephalans. And these are mainly so-called sand crabs. And there is an evidence for a food shift or shift in the food preferences in the sea otters because of the availability of the prey. In other words, because of the depletion of the other resources of the food in really destroyed marine environment along the American coast, the sea otters, they have less and less chances to find the proper food. So basically the population is slowly shifting towards the alternative food that was not eaten before by the sea otters. And this includes the sand crabs on the beaches. And these are the important hosts for acanthocephalans. So again, it's an ecological shift. It's a shift in the trophic web that leads to changes in the transmission pattern of the parasite. And this is something that we commonly see in the emergence of the situation of the parasitic diseases of so-called foodborne or food web transmitted parasitic diseases. That change of the ecology of the changes in the ecosystem leads to changes in the particular species ecology, which leads to a new contact between the hosts and artificial aberrant infections. And this is another cause that is driving the population of the sea otters down. So, and the case of Sarcocystis neurona already took us to second group of reasons or causes of the emerging infectious diseases, and it's a movement of the pathogens. We can conclude that the pathogens can move differently. They can move with their hosts, they can move with their intermediate parathenic vectors, they can move alone. One of the nicest examples of, again, textbook examples, dramatic examples of pathogen movement is West Nile fever virus epidemics in the United States. You know a lot about the West Nile fever, and you know about the issues associated with this and mosquitoes and birds in Italy. But the story was in North America is a little bit different, because the West Nile fever virus is known West Nile because it originates from Uganda. It was described in Africa ages ago. Africa is on the migratory route for the birds, so there is no surprise that the virus was always being present in some way in European continent, because our birds are going down there for overwintering. So, and on the way back, they bring the virus. But very surprisingly, the virus was found in 1999 in the US territory. I remember that from the TV, from the news in that time. I was already keen infectologist or parasitologist. So the news were full of birds dying in the Central Park and people being sick and the horses dying. So that was this year. The year later, the virus was known or was detected in much larger part of the eastern United States. The year later, it was almost half of the United States, and a year later, it was almost whole United States. So it took 4 years for this new virus circulating mainly among the birds and mosquitoes to colonize whole North America, which shows first how fast this is, and second, it shows how helpless we are stopping it. Basically, we cannot stop that. Yeah, we only can react, and it's not that pessimistic. Because of our knowledge, we can react, and this is what happened in the US. The vaccine was developed, the new ways to protect people and animals from the mosquitoes were developed, the new methods of the diagnostics were developed, and since then, the Americans, they learned to live with West Nile fever virus. And to be very honest, the same we should do in Europe. Introduction of new mosquito species is changing the epidemiology of this. It's basically a wildlife disease, nothing more, nothing less. But having a new vector, which is Aedes albopictus, is changing the pattern of the transmission. It's changing the importance, and suddenly the birds living in our environment are representing a threat for us through the mosquitoes. And this is why we see growing number of the human cases. This is why we see those summer epidemics. So it's not epidemic, but at least clusters of cases in summer. So far in Italy, but it will probably move north as a result of the pathogen movement associated with the climatic change. Back to the textbooks, we still have time. Avian malaria in Hawaii. Really, West Nile is not that much contributing to the species extinction. And because it's not that much pathogenic for the birds, and at the continental level, the bird population is able to adapt. So I'm not aware about any avian species that got extinct in the North America because of introduction of West Nile. The different story is avian malaria. It's also in USA, but in this case in Hawaii. And it's a really well-known case of collapse of the avian population because of introduction of new pathogens. If you look at Hawaii in this perspective, you see how isolated it is. It's one of the most remote parts of the world, really being thousands of kilometers from other continental land masses. Thanks to that, very rich, diversified avifauna evolved, endemic avifauna evolved in Hawaii, including mainly so-called honey creepers, but also any other birds. And many of them are extinct. Many of them are heavily endangered because of complex of factors, including the ecological changes, habitat destruction, including introduction of new predators, cats, ferrets, mongoose, dogs, pigs. All these are predators having direct impact on these birds, but also introducing the diseases. When we look at Hawaii, and basically other island ecosystem really mirror this situation, as part of the global change, we see the multiple invasions of non-native species. And for me, that was really shocking to read some years ago that 150 avian species were introduced to Hawaii. That's a massive, Hawaii, these are small islands, if you will. been there, or if you will go there, these are small islands, little islands, basically. There is 150 species introduced. And at least 50 of them, nowadays probably more, are reaching high population densities. So this creates the tension as such, or the pressure. But in the same time when the avian introduction started, also the vectors of diseases were introduced, specifically Culex quincke fasciatus. It's known there from 1826, again showing that the emerging diseases and emerging problems are not that recent. It was here always, not always, but since humans really colonized the world and start moving around, the diseases started to spread. And a few years later, also avian malaria was introduced to Hawaii together with the invasive bird species or introduced bird species. And the first cases of fatal avian malaria were observed as early as 1903 among the wild birds. So it's more than 100 years the Hawaiian birds are dying because of avian malaria caused by plasmodia introduced to them. And as a result, several species got extinct totally. So if we learn something from that, the introduced diseases in mainly naive, isolated marine island or oceanic island ecosystems, they can quite commonly show, or they find unprepared population of the birds. And then they reach really, or they cause quite high mortalities, with really having together with further stressors, having the impact on that. We can ask, okay, that was Hawaii. Is this happening also on the other islands? I mean, the oceanic islands, I don't speak about Mediterranean because these are islands too close to the continents. And we can say, yes, it is. The same, though smaller epidemics or impact of malaria, introduced malaria on native birds. We see or was documented in Galapagos, New Zealand, French Polynesia, Philippines. So basically everywhere where the endemic birds are living without previous contact with Plasmodium, they developed avian malaria. And if you look back to the captivity, we can see also the opposite trend, which is malaria, which is well demonstrated in malaria in the zoo. Or basically the same trend, but in different environment. Oh, no, it's not the same. It's the opposite. When the sensitive host is being introduced into the environment where the pathogen persists. So again, this is a movement, but in this case, it's a movement of the host. And these are the penguins in the zoological garden. So basically in any of the zoological gardens that are showing the penguins in the outdoor exhibition, we see the transmission of Plasmodium between the local avifauna, which is widespread or common, abundant in any zoo, the common European birds that are reservoir of the common Plasmodium species, which is not pathogenic for them. And the mosquitoes are responsible for transmission to penguins, which keep dying. And basically any zoo having the colonies of penguins outdoor is facing the same issues. Yeah. So and the reason is because like evolutionary, the penguins, they originate from the environment where is no malaria present. So they don't have that many inherited mechanisms how to cope with the disease. And the stories about mortalities among the penguins are everywhere. In UK, huge issues in many zoos. In the United States, people work in that. And in the Czech Republic, we have quite an experience with malaria in different groups of penguins. What are the solutions? One of the solutions is to show the penguins in indoor exhibitions, which is done mainly for the Arctic species. The problem is that it's expensive. Yeah, only few zoos can really afford having the exhibition of this costs billions, basically millions. Yeah. So here the penguins are protected. By more frequently, the penguins are shown in the outdoor exhibitions like this. And basically there is no way really to protect them, because you cannot really stop infection or Plasmodium exchange between them and the wildlife. Sorry, yeah. And which is taking us back to the zoo. The zoo is artificial environment, and moving of different animals of different origin, putting them together creates really really, as I said before, environment for unusual transmission. This is very aberrant infection. The penguin is not supposed having malaria because it's not supposed to live in the Central Europe or in Italy in a zoo. It's supposed to be in South America or extreme corner, south end of Africa and so on, in very different environment. And a story from the zoo, like showing another aspect of this, artificial contacts of hosts, is a story of anteaters, tamando anteaters from the zoo I work with, where we imported these very nice animals from Central America. And at the beginning, the situation was quite promising. The anteaters started to breed. We had first baby born and so on. And after a first relatively optimistic period of a couple of months where everyone was excited by new tamandua that came, we saw the mortality. First one animal died, then the another animal died. Female got sick. And it was revealed by a series of examinations that basically these animals are infected by gongylonema, which is a nematode parasitizing the oral cavity of them. So it was the first step. Fine, this is the diagnostics. It showed us a little bit the direction where to go with the treatment. But this is not yet the level at which we can propose some management. Yeah, we know that gongylonema is a parasite which is in the wild, but also in captive animals, transmitted by cockroaches. Blatella germanica, the common, I think it's called German cockroach in English, the common synanthropic insects living all around the world, basically in every zoo. I probably can, or I'm guessing that in every zoo there is strong population of Blatella. And this is very potent intermediate host for this nematode. So it was clear from when we identified the worm into the genus, it was clear that yes, there is some population of synanthropic insects behind it. But still, it didn't show yet the source of the infection. So we started to examine the animals, and we revealed that the South American primates living in the same exhibition together with the anteaters are the beasts or are the source of the infection. Shedding gongylonema, having the infection of the oral cavity, but in very low level. Shedding the eggs of gongylonema to the environment, where the cockroaches got infected, and apparently the anteaters are then getting infected by selective catching the cockroaches, reaching much higher infection intensities than the monkeys. So this was already a hint saying, okay, showing clearly, yes, we have to do two things. We have to separate the anteaters to the environment where are no cockroaches. It was the emergency move into the new enclosure. We have to start thinking about deworming of the original infection source. But it's still not the end, because all this is happening in some building full of other animals and full of the cockroaches. So basically we started and this is the management, yeah. We started to systematically examine one enclosure after another, which meant to examine, I can guess, few hundreds of fecal samples to show how the infection pattern is, because this pavilion is inhabited by totally different groups of animals, several species of primates, rodents, armadillos, sloths, and so on. And to really understand what's going on, all these animals were examined within a couple of months. And finally, we were able to prove, yes, that's not present in whole the collection. This parasite probably really circulates only from this particle monkey species to the anteaters, causing the mortality on the anteaters, and give us the tools to really, at least to break the transmission. We cannot really destroy the population of cockroaches. It's technically impossible. But we can start in breaking the connections, as I said before, separating the anteaters from the cockroaches, separated the anteaters from the environment contaminated by the monkeys. And finally, we are reaching the point where we can really focus on the dehelmintization of the anteater, which is already negative, but also to start the dehelmintization of monkeys. And we know that we have to focus only on these few that are infected. And this is what I understand under the term management. And this is something which is really complex issue. It takes months and months, and it's costly. It really costs money. And not of every zoological garden is able to afford this or is willing to invest this. And this is what is causing the complicated environment that I spoke about. Guys, tell me how much time we have. It's 5. Can I still continue, or you are almost dying at the opposite side?
Theoretically, you have other 30 minutes, but let's see if they are still alive.
Are you
I'm
still
alive.
alive?
Yeah, I'm alive.
Okay, so I will continue, let's say 15 minutes, then we have a few minutes for the discussion, and then I should drive home anyway. Yeah, so 15 minutes more, I promise, not more.
Okay.
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 Thank you for your patience. Anyway, so let's continue with the emerging diseases caused by movement and the translocation. And we look a little bit to the domestic animals, because also the domestic animals are, of course, being transported all around the world. We have 1 billion dogs living on the planet. Part of these dogs are traveling. If you look to the environment where the dogs travel the most, it's definitely Europe. Us Europeans, we are crazy in moving the dogs. They don't do it by themselves. No, we are shipping, taking our dogs from holidays, buying the dogs across the continents and so on. And this, of course, has impact on the disease transmission pattern. And because the dog is nowadays a dominant predator on the planet, 1 billion dogs, it's a massive population, which means also it's a massive reservoir of the infection. And what's important and what's not, in my opinion, generally recognized in Europe is that 80% of this dog population, or it's estimated 80% of the billion of dogs, are free-roaming dogs, which are the dogs which can easily acquire the diseases and spread the diseases to other dogs, to humans, or to wildlife. So it's one of the most important driver or reservoir of the infections globally. I like the dogs. I have the dogs. I'm not against the dogs, but this is the reality. We have 1 billion dogs on the planet. How many wolves we have on the planet? How many jackals we have? How many lions? Nothing. But the dogs are all around. And the most typical example of the infectious diseases that are spread by dogs and that are having really extremely detrimental effect or impact on the populations of carnivores globally is canine distemper. It's a virus that all of you know well, a morbillivirus, typically associated with dog population, but highly pathogenic for many other things like the pinnipeds, mainly the seals, African wild dogs, Ethiopian wolves, but also the lions, which is quite interesting because canine distemper was originally thought to be only in canine hosts, but in Africa, it was repeatedly clinically involved in the disease of lions, mainly in Serengeti. And if we stay with the livestock and the morbilliviruses, another not really well recognized, but extremely severe and important emerging diseases that happened twice was the PPR infection in Saiga antelope. You probably heard in your biology classes about the PPR. It's a viral disease associated with small livestock. Peste de petit ruminant is the full name of the virus. This is why PPR. But it happened twice in Central Asia that the infectious disease seriously hit the Saiga antelope population. You possibly know Saiga, it's a mid-size, it's a probably size of a roe deer or little sheep, size of antelope that was very abundant in the Central Asian steppes and semi-deserts. And it was one of the most distributed and most important herbivores in this ecosystem. In 2017, really mass mortality was recorded in Mongolia, and where the population was decimated to 20%. So 80% of Saigas died within 1 year, are the estimates. And unfortunately, very similar epidemics, but by different pathogen, in that case by Pasteurella, happened in 2015, just 2 years earlier in Kazakhstan, when they say that 200,000 antelopes died. So, and Saiga is a protected antelope. It lives in the national parks, and it's being protected in some way from the Second World War. So there is basically 50, 70 years of the conservation of this quite important animal of Central Asia. And within 2 years, from 2015 and 2017, most of this war was lost because of transmission of the disease from livestock. So it's serious. And of course, it's even more serious because if you imagine that the saigas as wild herbivores are, of course, important source of the food for the populations of endangered and really protected wild carnivores, like this is a snow leopard, but also some other. The reduction, 80% reduction of the population of this food item can have really unpredictable negative consequences for the populations of the carnivores. And this is something what now people are studying in Mongolia. This is something that we will wait for what will happen in the environment, which is now saiga-free. Are these carnivores still able to find enough food? Logically, they can turn more towards the livestock, which would definitely increase the human-wildlife tension in the area and maybe will lead to more persecution on these animals, driving also them to the extinction or to the edge of extinction and so on. So it's quite sad. And it's really huge question behind, and there are many articles on that. That was really big. In past years, it was big. The interest was a little bit slowed down in 2019 by COVID. But still, people keep working on that. People keep publishing on that. So if you want to follow the story, there is a lot of resources on the internet, but it's huge. Yeah. But the dogs are also example of other disease, which is very close to you in Italy, canine thalassiosis. Even though we consider that being a dog-associated disease, finally, it's not that dog-associated nowadays in Europe. Probably no need to introduce thalassia to you because you know it better than people here in the Czech Republic. Vector-borne spiroid nematode transmitted by photica flies, carnivores, and so on, detectable usually under the lid by direct observation and relatively easy to remove, relatively easy to treat. If you are aware that you have to search for that, if you are not really looking for that, you will never find it. But if you try, you find it, as you see here, not difficult. The worm itself was first detected or originates for sure from East Asia. It's well known for decades and decades and decades in East Asia. But suddenly, for unknown reasons, it was never documented as far as I know how it has happened. In 1989, it was described in Italy. And then it started moving through Europe, reaching most of the Europe in 2018. In that year, thalassia came also to the Czech Republic. So that's clearly an emerging disease, clearly associated with the dogs, because the dog is only carnivore that travels that far. No other carnivores travels between Europe and UK, or cats as well, but they are not important in this case, and so on. So only the dogs, only moving with the humans, were responsible for this massive spread. But what happened then, and it's well documented, for example, in Romania by team of Andy Mihalka, my friend and colleague, that from the dogs, infection spills over to foxes, finding perfect population of already very abundant red fox across the Europe. And then the infection starts spreading in the fox population. And for example, from Romania, from where we have good data, we know that 50% of foxes are infected. So it's massive again. So whole country basically is full of thalassia callipeda. And then logically from the foxes, the infection is spilling over again and again and again on the permanent matter to dogs. So it's not a canine disease, it's a wildlife disease nowadays in Europe. But the driver of the infection was movement of domestic animals and spread, even though we consider as veterinarians, we consider thalassia a pathogen of the dogs because we treat our dogs and our own dogs can get infected. but it's a wildlife disease that was imported or introduced by the dogs. And it's one of the strongest examples because we have good data from that. Thanks to Italian research team, we have really good data, and Romanian later on, we have good data on this. For example, from the Czech Republic, we still are at the beginning of that. So we are more or less probably facing the silent phase where the parasite is being slowly circulating among the fox population. But then we can expect in coming years, similar to Romania, for example, we can expect a boom of the infection in the dogs. When the fox get infected, they live everywhere. The infection will jump to the dogs in huge frequency, probably. Yeah, and here it opens quite nicely. This case is the previous one, but also this one, the question of the role of the veterinarians. Yeah, so in those parasites that are primarily at the beginning associated with the domestic animals, we have a range of opportunities to control them. We can control the movement of the domestic animals first. We can control the contact of domestic animals with wildlife, which is nowadays being finally implemented around some populations of the wild carnivores. Yeah, so simply we are too much pro-dog oriented society. And if we want to keep some sensitive wildlife safe from the dog transmitted diseases, we should make some barriers in our behavior, in the rules. So simply it's not always a good idea to take your dog for a walk to the environment in which the endangered carnivores are living. This was the case of Mediterranean seals, for example, or the other endangered seals around the world. The dogs bringing the canine distemper to their groups. And we also have, as veterinarians, we have a lot of therapeutic and prophylactic measures to prevent this unwanted spread and to protect the domestic animals, but also logically to protect the wildlife. So it's very nicely connected, and this is what we can consider a management. We are not speaking about individual dog. We are speaking about the dog population to be treated properly specifically in case when it travels. But this is not happening. Thousands of dogs are nowadays moved across Europe, from Romania, from Greece, from Spain, as a part of so-called, I have to say, animal welfare activities. And we are bringing the dogs with the diseases, with Thalassia, with Leishmania, with Babesia, with Ehrlichia, Birofilaria immitis. All this is happening in massively in Europe. And we are just facing that. We are observing that. We take notes and write publications on that. But still nothing is truly happening. And in some situations, this can have impact on wildlife, which is something that at least me, I really don't wish to see. And this is responsibility of vets, no one else. Veterinarians and the owners. And the communication, proper communication of veterinarians and the owners, and communications of the issue also. Not only that, yes, Thalassia, it's an issue for your dog. Sorry for that. Yeah, the dog have inflammation of the eye. Yes, that's sad. But what's more sad that your dog brings the Thalassia to a population of wildlife, and it's Asian worm, so it's not supposed to be in Europe. Unfortunately, this is already happening. Yeah, and I think that's it. I have a few more diseases, but I think we can stop at this stage to give you some space for the questions. So, guys, please, if you have any comments, questions, disagreements with my sometimes maybe a little bit extreme standpoints or positions, so please feel free to discuss. I'm here. And also in the talk, there was a email address of me. So if you wish to discuss something in the future or in person, because I'm coming in less than 1 month or almost exactly in 1 month, I'm coming to Sardinia. So we can see each other, discuss more. So questions, at least 1 or 2, just to know that you are alive.
Thanks, David. I would like to ask you if you already spoken about your activity with CHITAC and the possibility for students to go there.
Yeah, I will show that. I have I don't have many slides. I have showed 1 slide in the beginning.
Yeah,
yes, I can introduce that. Yeah, but it's gradually changing a little bit. But I'll tell you the story. We did some research, and we still do some research in universities with Somaliland, which is the northern part of Somalia, the autonomous part of Somalia, where is a huge shelter for cheetah. You know, cheetah, the African carnivore, gepardo, right?
Yes.
Gepardo in Italian.
Yes.
There is quite nice opportunity for volunteers to get involved in the work of cheetah shelter. Since the past 5 years, when I was more involved there, the situation improved a lot there. So there is already quite nice center managed by Cheetah Conservation Fund. They still accept the they accept the volunteers. Just for you to know, there is about 100 cheetah already. When I had this lecture a couple of years ago, I said there was 40, then it was 60, then there is, and now we are reaching they are reaching 100 cheetah in there. These are animals from the illegal trade, so they are captive cheetah. Now the center got new land with quite exciting opportunities, and they accept volunteers. Nowadays, the application doesn't go through me because originally the volunteer program was started by us. But now the Cheetah Conservation Fund, they have their own web link and like application way. So those of you that are truly interested to have hands-on experience with 100 of semi-wild African carnivores, the project of Cheetah Conservation Fund in Somalia, or Somaliland, it's the northern part of Somalia, is really interesting, and I definitely recommend it. In past years, we sent maybe 15, 20 students, maybe 15 students there, and they all, mainly students of veterinary medicine, but they all came really happy after spending like unsatisfied after spending 1 month of the daily work with 150, doesn't matter, cheetah. It's enormous. Yeah. So if you wish in the future to work with wildlife and wild carnivores, if you have good experience, either you can easily Google it, Cheetah Conservation Fund, or you can contact me and I can manage the contact of you to them. I just want to remind about saying that Cheetah Conservation Fund has 2 shelters. One is in South Africa, in Namibia, which is quite expensive, well-developed, and a little bit posh conservation center. But if you really want to work with animals, it's much better to go to the new center, which is in Somalia, the one I spoke about. So if you have any questions, feel free to ask me. If any one of you, and most of you probably do, love big cats, this is an opportunity to have hands-on experience.
Okay, thanks.
I have a question about what we just were talking about the program. What kind of activity do the volunteer do in the preservation center?
Depends on their level, depends on their knowledge and so on, but it's daily care, which means basically all activities, starting with preparation of the food, cleaning, playing with the cheetah, because many of them are babies that deserve some activities, petting them, treating them. There is 1 or 2 veterinarians, I mean good veterinarians, also assisting the veterinarians in the daily things. There are not that many surgeries done, but if there is some accident or something, even this can happen. But usually it's basically the small veterinary activities, vaccination, feeding, supplements, enrichment. So like just whatever you can imagine. If you have 100 cheetah, what you can do with them. So this is the work for the for the volunteers. So it's quite intense, to be honest. It's 12 hours a day almost work with cats.
Okay, thank you.
Welcome. And it's safe. The country itself, it's safe. Yeah, I just came from there 4 weeks ago, and it's better and better. I really love the country, to be honest. Yeah. So if you want to see something interesting animals in very interesting, though a bit extreme environment, Somaliland is the place to go. And anyway, you Italians have really deep roots to Somalia, so it's your second homeland almost. Yeah. You can come again to Somalia.
Yeah.
But Somaliland, like the northern part, was British originally. This is why it is a little bit independent, because it used to be a British mandate territory, while the rest of the Somalia was mainly Italian, historically. So they speak good English, but they still have pasta in there, so good combination.
Okay. So, David, I think that it's time to thank you. I have a
There is a question in the chat.
Oh, yes.
Sorry, in the chat. I don't see it. Where is it? It's here. Sorry, opening. Presentation. PPR disease in West Africa. What I know, yes, but it seems that in Africa, the wild ungulates can be a reservoir, but it doesn't have that huge impact on their population because it's well possible that the virus originates from Africa. Yeah, so I'm not really following that much in livestock. So for sure, it's present in Africa. There is a huge FAO-driven campaign for vaccination of goats and sheep against PPR in Africa. But the data from wildlife are not big, but it's a really good question because the same question, in fact, we were asking ourselves in the PPR meeting and FAO in Rome, because there is a global ongoing campaign for eradication of PPR in livestock. But if we consider that it's well possible that specifically in Africa, there is a lot of wildlife reservoirs, then logically the eradication campaign should fail. And this is now a really big discussion, whether it's reachable, the point that we eradicate the PPR. Because the FAO, as the leading agency in this, was really motivated by eradication of the rinderpest. You probably know that a couple of years ago, the other morbillivirus causing the infection in cattle was successfully globally eradicated. It's one of the few. And having this, FAO felt that they have mandate to say, okay, we eradicate also PPR, but it's present in the wildlife and this probably makes it not doable. So this is what I know about Africa and West Africa. Fine. That's
Okay.
it. Okay. So it's getting late. I still should drive 200 kilometers back home. So I'm in Prague today. So guys, have a nice afternoon. Thank you very much for your time. And
Thank you.
I will see some of you possibly in a month.
Thank you for the presentation.
Yes. So for PhD students, if you need the recording, I will share with wildlife managers, students, the file. If you need it, just send me an email. Okay. Grazie, David. Thank you again.
Okay, thank you.
Thank you. Goodbye.
Bye bye.
Thank you. Goodbye.